Today we will discuss about whether this lactate that are produced in the pancreatic cancer cells helps in the polarization of the macrophages or not. So the results that are presented here are based on the in vitro experiments that are done to check whether the lactate can mediate polarization of macrophages or not. In the previous video we have find out that this Warburg effect cause pancreatic cancers to produce lactate. At the same time, it will also leads to this aerobic glycosylations. So in this video, they check or in this results figure, they have explained whether the lactate helps in the polarization of the macrophages or not. So what they did is they take the conditioned media from the cells. They take the conditioned media from the cells that has this Si silencing VCAM1 and or silencing RNA VCAM1 or silencing RNA for the control with this quercetin, and quercetin is known for inhibiting this traditional lactate mediators such as MCT1 and MCT2. So in this case, what they have find out in the panel A is that when they use only this control silencing RNA that does not silence VCAM1, they can see that the cells are less rounded shape. However, when they use this silencing RNA against VCAM1 conditioned media from the pancreatic cancers that uh, where this silencing VCAM1 RNA is used, so those conditioned media will cause more rounded like structure formations of the macrophages. And when they use this silencing RNA for the control with these quercetins, they also see that rounded structures are there compared to the control case where only the conditioned media from the pancreatic cancer cells, where pancreatic cancer cells are introduced by this silencing RNA, the control silencing RNA that does not silence VCAM1 or the lactate productions in other terms. And similar things they have observed for the capen2 cell lines also, where they have seen the more rounded structures formations whenever they are silencing VCAM1 RNA is used or this quercetin is used, indicating that this lactate formations is the this lactate inhibitions will cause this kind of rounded structure formation of the macrophages. And when they did the RNA-seq analysis or mRNA levels, when they check the mRNA levels in these samples, what they have find out is that the markers for the M1 macrophages are reduced in these rounded structures formations compared to the control case, indicating that this lactate which are formed, this or soluble lactates which are formed from the pancreatic tumor cells helps in the polarization of the macrophages, or whenever this VCAM1 is been silenced or this lactate formations are been inhibited using quercetin, they see that reductions in the mRNA levels. And they check the different markers for the M1 macrophages, for the activated macrophages such as CD206, CD163, fibronectin, CCL18, CCL22, interleukin 10, and they have find out that all these markers are reduced in the pancreas cells and also in the capen1 cells. And when they check the cytokines concentrations, what they have find out cytokines concentrations are also reduced. So cytokines concentrations such as CCL18, CCL22 and interleukin 10s, these are also helps in the activations of the macrophages. So these are also reduced in the pancreas cells and also in the capen2 cells. That can be seen here in the results. And after validating that the lactate formations affects the polarization of the macrophages, they wanted to check the dose dependent effect on the lactate of the lactate on the polarization of the macrophages. So what they have find out is that with this increase in the dose of the lactate, they can see that increase in the roundness of the cells of the macrophages. This indicates that the lactate has a dose dependent effect. Lactate has a dose dependent effect on the lactate has a dose dependent effect. Let me correct this thing. I have said something wrong. So as the lactate concentrations are increases, we can see that there are more spherical forms macrophages are observed. For example, in the control case, we can see that this seems to be the circular shape, but with the increase in the concentration of lactate, there are less round formations of the macrophages can be seen. And here in the 25 millimolars, we can see that very less round formation structures or different structures of the macrophages can be seen, but these are not round shaped. And when they check the mRNA levels, what they have find out is that the marker or the markers for the macrophages, activated macrophages, are increased. That can be seen here, and these increase are relative or in respect to the increase in the lactate concentrations that can be seen here. So this indicates that with increase in the mRNA levels, with increase in the lactate concentrations, there is increase in the activations of the increase in the polarizations or activations of the macrophages. And when they check the cytokine concentrations, they see the cytokine concentrations are also increased with increase in the lactate concentration, indicating that the more lactates are, more cytokines are formed or released to convert macrophages, inactivated macrophages or M0 macrophages to the activated macrophages or M1 macrophages. With the increase in the lactate concentrations. So I hope you got the idea that lactate in the pancreatic cancer cells helps or mediates the polarization of the macrophages or the activations of the macrophages from M0 stage to M1 stage. If you need to understand more about these experimental results in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.